Welcome to the 26th Distinguished Statistician Colloquium, sponsored by Pfizer, ASA, and UConn. We are grateful for our sponsors' generous support. I am Dr. Deepak Dey, UConn Board of Trustees Distinguished Professor of Statistics. Thank you all for joining us today. I am honored to welcome you and provide an orientation for you. My colleague, Dr. Haim Bar, will guide us through the later portion of this program, and you will see him later. Today, we are here to celebrate the distinguished career of Dr. Peter Bickel from the Department of Statistics at the University of California, Berkeley. We will be joined later also by Dr. Lisa Levina from the University of Michigan, Ann Arbor, Dr. Purna Sarkar from the University of Texas, Austin, as well as Dr. Rachel Wang from the University of Sydney. We encourage you to ask questions as they come to you using the Questions tab on the left next to the Program Agenda tab. We will compile your questions for our interviewers to ask during the interview portion. One lucky question answer will win a commemorative tote bag, and the winner will be notified tomorrow via email. If you have any tech issues or questions, you may submit them at the Questions tab as well, or email conferences@uconn.edu. But first, let's have a little Peter Bickel trivia. Shortly, you will see a poll question pop up on your screen. If you don't see the poll, go to the tab that says Live Polling. How many academic residents does Peter Bickel have? A, under 300, B, between 300 and 500, C, between 500 and 700, and D, over 700. Let us know what you think the answer is, and we look forward to sharing the results after the keynote address. At this point, I am delighted to invite my colleague, Dr. Nai-T Ting, who has been kind enough to record a warm introduction to Peter's keynote for us. Dr. Ting recorded his video before we learned the good news that Dr. Rachel Wang should be joining the team of interviewers as well. We are delighted she could be part of this. Dr. Ting.
Good afternoon. Welcome to the 26th Distinguished Statistician Colloquium. My name is Nai-T Ting. It's a great honor for me to be here to open up this very important event today. Now, the Department of Statistics at University of Connecticut is honored to announce the 26th Distinguished Statistician Colloquium. And actually, this colloquium was co-sponsored by American Statistical Association, Pfizer, and Department of Statistics at University of Connecticut. It turned out that I had the honor to be associated with all these three important organizations. I used to work at Pfizer between 1987 and 2009, so I was a past Pfizer employee. And I'm a fellow of American Statistical Association, and I'm also a lifetime member of ASA. And I'm also an adjunct professor at Department of Statistics at UConn. So it's a great honor for me to open up this important event today. As a matter of fact, today I also represent another important organization, which is New England Statistical Society. As many of you know, starting 2017, there is New England Statistical Society that was officially formulated. And of course, for over 30 years, there's a New England Statistical Symposium. So there's a long history of this organization. The Pfizer Colloquium series ranges from 1978 until 2012, and then was renewed in 2018. The Colloquium series featured C.R. Rao, Bradley Efron, D.R. Cox, Grace Waba, and many more. For a complete list, please visit the website. The purpose of the colloquium is to provide a forum for distinguished statisticians to share and disseminate their unique perspective and work in the theory and/or application of statistics. Starting from 2018, the series has been co-sponsored by Pfizer, the American Statistical Association, and the Department of Statistics at the University of Connecticut. The 26th Pfizer ASA Yukon Distinguished Statistician Colloquium is scheduled on Wednesday, April 21st, 2021, 3 to 5 p.m. through webinar. This year's speaker will be Professor Peter Bickel from the Department of Statistics, University of California at Berkeley. The title of his presentation will be 4 Excursions in Genomics. The colloquium will be followed by a discussion with Lisa Lavina from the University of Michigan, Ann Arbor, and Pralamarita Sarkar from the University of Texas, Austin. So without further ado, let me introduce our speaker today, Professor Bickel.
Pfizer ASA Yukon Colloquium. Since Pfizer's involved, I decided to speak about a few things that I did in biology, although this of course not been the main thrust of my work. And so let me start out with these 4 excursions in genomics. Oops. So here's an outline. I'm going to talk about the ENCODE project, which was the biggest thing that I was involved in connection with biology. And then a couple of papers connected with that. One, which was a direct contribution, which actually seems to have been still exist in the pipeline, and a general contribution, which was more of a paper, but I hope still useful. Then I'll talk about network models in general, very briefly, to introduce people, and then talk about 2 papers, which I did with each one with one of my collaborators, who's going to be speaking later, and then we'll proceed. Okay, so this is really glitzy. It's not my slides. They were made by Mitch Snyder, who's a big genomicist. And telling you about the ENCODE project, there were, as you can see, 3 phases, and the original goal was the humble one of annotating functionally every element of the human genome. Of course, this went on for years and years and years, and at this point, at the point that I quit at least, and it's continued, they were satisfied with biologically annotating every element of the human genome, biochemically. But in the process, they generated a lot of useful data. And that's where we had some share. But just to give you an idea of the effort, there were 1,700 assays, as you see, and this is as of 2015, of all types. By now, there are many more of more sophisticated types. And at one time, at least, there were 350 people all over the world working in this effort. And for a while, we were fortunately, by accident, the only statistics group associated with it. A consequence of this study, which was that, and of course a lot of work in between, was the number of protein coding genes actually that people believed went down. They discovered thousands of non-coding genes and much more potential regulatory DNA, but only potentially so. By now, of course, much more of this has been validated. So here's some contributions of our group. I chose two papers, and there were, I think, a total of 4 or 5. One, both appeared in the Annals of Applied Statistics, and as I said, one of them at least, well, both involved a number of collaborators at Berkeley, and one who's now at Penn. So the first paper was about, I'd like to call it well, it became known as the IDR, the irreproducible discovery rate. And why that's so, I will become clear. Essentially, all of these assays were going on. One of the principal assays that were going on were ChIP-seq experiments. And they, or one person in particular, Ewan Burney, at the very beginning, specified that they should do at least 2 copies of the same assay, so that to have some check on quality. And that's where we got involved. So what they wanted was to uniformly process data from multiple sources, compare the performance of different algorithms, select the best one to process the data. Even then, there were at least a dozen algorithms for reading ChIP-seq data, and to select the peaks using a uniform criterion across all data sets. The peaks is what you're looking at, the biologically meaningful peaks. Well, no ground truth is available, of course. The peak calls depended on the parameter settings of the individual algorithms. They all had somewhat different significance scores. What these significance scores meant was not terribly clear, and they were certainly not calibrated with each other. And so the goal, our request, was to try and calibrate these, if you want. And the idea is that replicates help in the following sense, that genuine signals should be reproducible across replicates. And that's what we can check. And so that's what we proposed. Now, the naive solution, the very most naive solution was, you have, let's say, these 2 replicates. And so for each position in the genome, you have a pair of numbers, one from each replicate. And you tell yourself the story that either they're real, right? They're really measuring the same thing, in which case they come from some bivariate Gaussian, which is a story, or they're garbage, which means that they're pretending to come from independent Gaussians. So this, in fact, roughly speaking, did correspond to what one wanted, but there was something really wrong with it. And let me tell you, well, first of all, let me tell you how one would analyze this. I think many people are familiar with this. You would use the EM algorithm, would be one way of doing it. That is, this is a mixture model. You initially guess which component of the mixture each observation comes from. Then, given an initial theta, you can compute very easily the new theta, repeat the process, relabeling the observations in the new way, and continue. But there was absolutely no reason to expect Gaussianity. So what we went to was a copula model. So what's a copula model? It's basically a model which recognizes the fact that the X and Y measurements are not on any comparable scale necessarily. And so you assume, though, that there exists an unknown function, one on each, for each coordinate, intensity, which are smoothly increasing, and such that the inverse of that unknown function, right, in fact satisfies the Gaussian mixture hypothesis. And now you proceed as before, estimating H and identifiable parts of the parameter. Now what happens, of course, is originally you could, of course, you had means, variances, covariances, and so on. But after this transformation, which you don't know, all you really have is the difference of the means, the ratio of the variances, and the correlation. So here's the solution that we proposed. So there's a basic relation which we start with. So if you have the marginal CDF of one of the variables and the marginal CDF of the other variable, and theta is the parameter labeling everything which you want, then you can write down what these are. What you observe is in terms of the original variable CDFs. and you get a naive estimate of the transformation. Right? If you know theta, you have a naive estimate of the transformation. So pretend that that's the case, and you estimate it, as you see, by fj inverse of f hat j of x. F hat j of x is the observed marginal and the theta which you assume. And then what you do is you basically update, given that you know the theta, you estimate by the observations, if you want, by the inverses for that given theta of the ranks, the normalized ranks of your observations. So because that's all you really know. You only really know the ranks of the pairs of ranks of each pair of observations. And then you do the same thing as you do before, that is the EM algorithm, but you basically retransform the data each time. And then you iterate this to convergence, and you hope it converges, and you hope it converges to the correct thing. And most of the time it does, if you have reasonable starting points. And it worked reasonably well. That is, we could look at different algorithms. That's what this table shows. And it clearly shows that some algorithms are better than others in that they agree on a larger number of the high peaks. Right? And in fact, that algorithm was adopted. We even had a little departure by which we identified that they were doing something wrong in one of the assays because the results were inconsistent in a very poor way. Okay, so that's the story for this paper. There are a lot of details, and what's most important is that the algorithm was taken by Anshul Kundaye, who is now a quite well-known biologist, and put into a big pipeline which exists still and which is being maintained. And so now this is being used not only for ChIP-seq, but a lot of other data at the same time. And the irreproducible discovery rate, of course, corresponds to the stuff that you shouldn't be paying any attention to, right? It's irreproducible. The next paper, which I'll talk about very briefly, was a joint paper which was basically prompted by the following question. Again, you suppose you have two features on the genome, and you look at a chromosome, and then you see that these features, transcription start sites or binding sites or other things, overlap each other. And the question is the overlap by chance on the genome? Well, there's a problem, right? Because you don't have no idea of what the underlying probability is. And we've had the following formulation. We need to define expectation at random. The genome is highly structured, and so the analysis of feature interdependence must account for superficial structure. So expected at random we translate it as overlap between two feature sets bearing structure but under no biological constraints. And here's the model. You basically tell yourself that the genome is divided into stationary pieces, but you don't assume anything about the stationary mechanism, right? But it's stationary, and it's also strong mixing, which is saying something fairly reasonable. You have homogeneity within the blocks, more or less, of sequences of base pairs. And furthermore, base pairs which are very distant from each other have relatively little to do with each other. Now, there's a problem, which is that these segments are actually not, as we think of them, necessarily connected strips. The problem is that the genome is three-dimensional, and its parts will be touch each other, behave as if they were next to each other, which are in fact not next to each other. So these homogeneous strips are actually more or less, despite our hopes, impossible to identify just from the data, right? You basically have to rely on biology to tell you what's stationary, what's homogeneous. The nice thing about it is, as I'm here repeating what I said before, that distant things have little to do with each other. But if the model holds, it also applies to derived local features, right? So it doesn't just apply to every point, it applies to small segments of points around any point, which you can slide along the genome. And so we needed, basically, however, to somehow get the randomness of what we observed, the particular overlap of a pair of features. And what we decided to use, with some refinements which I won't talk about, is a block bootstrap. So essentially, you draw a subsample of a length much smaller than the length of the stationary thing that you're studying. You compute the statistic on the subsample. You do this many times, right? And that gives you an empirical distribution. The only trouble is that the empirical distribution is based on fewer observations than you actually have. But some theory tells you that, in fact, these things are all operating at scale 1/√n. So what you do is you take your distribution, you rescale it by the square root of n over m, or m over n, and then you pretend that that's your true distribution, and you can now compute confidence intervals, whatever, from this. So this is the theory that I was just talking about. And I think I'll just skip that. It's the justification for what I just described, working. And the issues, as I mentioned before, is choosing a method of segmentation, which you can only do by biology. And another issue is, how do you choose the block size for the bootstrap? And you have to do it using stability considerations. And then the other issue is segmentation, the size of the segments. And we have discussed some things. Okay, now I'm going into the realm which I think my auditors, commentators are more familiar with, which is two papers, one of which was done with Lisa and one of which was done with Rachel and others. So let me begin with the first one, which was done with I'm sorry, not Rachel. Yes, Rachel and Purna, in fact. Sorry, I forgot. So let me go on to that. So what's a network? And my definition broadly is that you have a representation of a set of data objects, people, computers, genes, etc., exhibiting relations between pairs, friendship, communication, regulation, whatever. Usually covariates are present, right? You have properties of the objects, educational, economic, locational. And then you also have properties of the relations, possibly, the strength and the type of relation. And of course, your interest is in some latent variables, features of this set of individuals, which you cannot observe directly. And in particular, we focused on community structure and parameters such as centrality values. We actually featured more on the community structure. Well, here's an example of a network, which I'm not going to talk about, but I like the picture. This was done by Mark Newman. And it's basically a food web in the Weddell Sea, which is way off in the Arctic. And basically it tells you, more or less, you have an edge between, the objects are, of course, species of animals, and you have an edge if one eats the other. There's actually a directed edge, but we ignore the directions. And I'll come back to that simply as an example of what I mean. So here's the formal representation. You have covariates, xi and xj, associated with the points forming a pair, potential pair, xij, which is a covariate having to do with the edge, possibly wij, which is the strength of the edge, and then the aij, which tells you basically if there is an edge or not. The graph is simply the set of vertices, the points, and the edges. And what you are interested in are the unknown features, this z, which could depend in principle on the whole graph. However, the probabilistic assumptions will come later. So for the Weddell Sea is actually, I put it down because you can see easily what kinds of things are involved. Here, Aij, as I said, does one or the other eat the other. And the covariates are the body mass, the indicator, whether I eats J or J eats I, right? And then what we're interested in, of course, is community membership. And communities here, fortunately, this is one of the things that makes ecology a nice subject for this study, is that there is ground truth, right? You have some idea of what the species are. But as I said, I'm not going to talk about that. Now, what about models? And I'm going to dwell on that briefly before launching into the papers themselves. So the basic and key assumption is that given the covariates and given the latent variables, the distribution of a pair of points of an Aij, of an entry in the adjacency matrix, depends only on the covariates associated with those points, and of course, in principle, the whole matrix of latent variables, but you can think of them as being associated each with one. So if you don't have a weight matrix, which we won't for these papers, what you have is that the conditional probability that Aij equals 1 is a function of the Xij's, the Z, which you don't know, and a parameter theta, other parameters theta. Now, there are two special cases, which I'll just talk about very quickly. One is the case where you have no dependence on anything. This is the famous Erdős-Renyi model. You just toss a coin to decide whether you have a line or not, an edge or not. And then next up, which have, and these two things have been studied more intensively than almost anything, mathematically certainly, block models, where basically, again, you have an edge formed through the probability which depends upon the membership of the two ends in communities. Okay. Now, how do you fit such models? Well, they're basically, I would say broadly, two types. One's based on the likelihood and spectral methods. And the likelihood ones are, you just write down the likelihood pretending that you actually know the identities of the communities, right? And then you basically optimize not only over the parameters which govern the model, which of course you could estimate if you knew what the communities are easily, but also, unfortunately, over the distribution of assignment to the communities. And that's where the problem comes. It's very slow. The other way is based on this observation. It's a kind of method of moments, that the expected value, the probability of having an edge given the community membership, is that matrix is of rank, which is the same as the number of communities. It's an n by n matrix because there are n vertices, but its rank is only equal to the number of communities. Therefore, you can, by a famous theorem, find the closest matrix of rank k to the matrix you start out with. That turns out to be determined by the spectral decomposition. And then you do the representation of the matrix A in terms of the basis of the top k eigenvectors, top k corresponding to the largest eigenvalues. And then you simply cluster, typically by k-means clustering. Unfortunately, as you can see, that has another problem, which is not really applicable when you have covariates. So what does it mean? It means that you basically have to some extent, tool each application to tool your models to some extent to the application. And the first application I would like to talk about is of the paper that I did with Rachel and Purna, having to do with things called TADs. So what are TADs? TAD stands for topologically associated domain. And the idea is that you try to find out what the three-dimensional structure of the genome is. Because, as I mentioned earlier, genomes, which are actually quite, the single chromosomes are I'm sorry, the string of base pairs is very, very long. And it has to be packed into a much shorter length, namely the chromosome. And that packing is done by forming tightly packed helices around a central core. And of course, what that means is that stretches of the genome, which are in fact, in terms of length along the string of the molecule, long, are actually quite close together. So knowing the three-dimensional structure is quite important. And each of the chromosomes is a single molecule. At least we know that. Okay, well, not so long ago, by our usual scales, but in fact, it's a remarkable discovery, which is that you could construct experiments by which you could determine parts of the genome which are close together, essentially in three-dimensional space. You can find contacts. You can find out for a pair of bases how likely they are, in some sense, to be together. And they could be together because they're very close on the line, or they could be together because they're folded closely together. Okay, so this leads to a network in the end. So here's the model. The model is that you observe an n by n matrix, which is thresholded to become an adjacency matrix. So the matrix originally tells you, what the strength of the association of the 2 points on the genome is, right? But you threshold that to create a 0-1 matrix. Now, there's also some bit of information which is available here, which is that it's known that the boundaries of these stretches, which are folded, this piece which is folded together, the end of the loops are things called CTCF sites. Of course, you could, as you saw from the picture, you could have loops within loops. Now, the CTCF sites are identifiable. So what you basically have in terms of data is this adjacency matrix, and also you know where the CTCF sites are, so that you have covariance. What you don't know, though, however, is whether the CTCF sites whether the CTCF site you're considering is the boundary of one of these domains. That's the latent variable. So it's in some ways a nicer problem than the general problem, because you know that the communities are strung out along a line. So you know that there are unknown points. You don't know how many, but let's say you pretend to know how many, which are genuinely boundaries of these topologically adjacent domains. And the communities are defined by consecutive members of a subset of the CTCF sites. So there's putting them in our framework. The probability of Aij equals 1 is a function of, if you knew what the boundary sites were, it's a probability, it's a function of that. And if not, it's just random. Now, you, in principle, could fit by likelihood. This is not quite as bad as the original likelihood problem, but it's bad enough because there are a lot of these guys. And so, in fact, variational likelihood, which I certainly will not describe, stochastic gradient descent, ascent or descent, whichever way, was used. But an algorithm was devised which worked. And although, of course, we could not establish ground truth, at least there's a weak test which basically says, well, how many histone sites of different types are there in your segment? If there are a lot, it's plausible that it is a TAD. And the method worked somewhat better than ad hoc approaches. Now, the next paper that I'd like to talk about is a quite recent one, which is just appeared online in JASA. This is joint work with Lisa, Purna, and not Rachel this time, but Lisa and Porn at least. and it's basically trying to think about what we call hierarchical block models. So what is a hierarchical block model? Well, you begin with believing that there's an underlying graph structure, which you don't know, but in which you could position the vertices. Or what, no, sorry, you position the communities, in fact. And you label them by nodes of the graph. There's one at the top, then two, then four, and so on. It's a binary tree. And you can imagine, for example, what at least in my mind, this mimicked a hierarchy where you have, there's the president of the company, and then there are the next level of executives, and these have other executives under them, and so on. This is not the application I'm going to talk about, but that's, I think, something to keep in mind. And what's, of course, of interest are communities at any level, in some sense. A community which would be, for example, everybody reporting to the 0-0 vice president, as opposed to community reporting to the 0-1 vice president, and so on. Well, I should mention, as you can see at the bottom, that this is actually binary trees were initially analyzed in a paper by Peel and Closet, earlier in papers by Newman and Closet, but they viewed it from quite a different point of view than we did. So, but I've told you a vertex structure, but I have not yet said anything about how the edges are placed, what the chances of edges are. So basically, you think of the tree as being of a depth D. D, depth in the usual sense. The number of communities is less than or equal to 2 to the D. In other words, you identify primitive communities with the leaves of the tree. And there are 2 to the D tree, leaves at the end. And then you have these other communities that we're interested in, which we call meta communities, which are all the leaves originating from a specific internal node. So now these communities are not like the communities in the block model because they're actually quite heterogeneous. Okay, and now the key is, how do you define an edge? Well, we define it in a rather natural way, which is that the chance of an edge depends upon the last common ancestor of the 2 leaves. Right? You go back in the tree and you find the origin, the first origin root to which both of these guys belong. And then you have a function of that, and it's the plausible assumption is that, at least for the models we were thinking of initially, is that this is decreasing. That is, you are much more likely to have an edge with somebody at your own level, a direct edge, than with somebody at a higher level. Okay. So the canonical algorithm is that you test a naive hypothesis. You begin with your graph, you test the naive hypothesis that the model is an Erdős-Rényi model. In fact, in other words, there's nothing there. All of the probabilities of edges are the same. If you reject that, if you accept that hypothesis, you stop, you quit. But if you reject it, you then fit again for each of the 2 remaining models. Now, one of these you may stop, the other one you may continue, and so on. And there's a sum stopping rule, which again is generic. So you create your tree, and once you've created your tree, then you can start to fit, and that's usually not a big problem. You can use spectral clustering or whatever. Okay, well, since I said that this was a talk about biological issues, this too has a biological application, which I'll discuss. Admittedly, there are many others, and in fact, the principal application of this in the literature was to collaborative networks. networks of collaborators. However, we had a biological application which I found very interesting. And this was due to Cohn Vandenberg, who was one of the members of one of the authors. So you want to cluster genes into biologically meaningful meta-communities, as I said, using some external measure to determine whether a pair of genes are related or not, have an edge between them. So the start was the data, and the data was a large corpus of papers from a broad area in biology linked to anemia. So anemia was a major term in each of the papers. And then you reduced to the genes appearing together in at least 2 papers. That went down to 1,500, I think 1,500 genes. But then on various grounds, genes which had little contact with anybody, we pruned it to a relatively small number of 140 genes. And then we applied this hierarchical algorithm. And you get this very pretty picture. It actually stops quite quickly, right? You have a total of 6 leaves only in the end, and a number of meta-communities. And these one could examine. One could then go and examine what did the genes in each of these meta-communities have in common. And it turned out that you could identify, for example, the community 1 involved cell activation, which is involved in immune responses. These all had to do with anemia. Another one was cell activation alone. A third one was response to cytokine. A fourth was DNA repair. Fifth was cytokine-mediated signaling, whatever all these things really mean. And oxygen transport. The thing I have to admit that struck me the most was that in, I forget which number community, a gene appeared which was a major hub, as it's known. That is, it had many, many genes which were connected to it. And guess what that was? That was the gene for sickle cell anemia. And sickle cell anemia is a rather unpleasant disease, which affects mainly people of African descent. It's actually a strange disease, which on the one hand is very unpleasant, it's very painful. On the other hand, it protects against malaria, or the gene protects against malaria. It has 2 different functions. But in any case, when one looked at the biological features in each of these communities, they could be explained quite reasonably. As usual, there are exceptions, there are things that you don't know about, but the majority view was fairly clear. And that's the end of the papers. So let me end with a picture of all the people involved in all of these papers at different times. Lisa, Purna, Rachel, down on the second row, Sharmadeep Bhattacharya, who's now at Oregon State, Shrimendra Mukherjee, who's at the Indian Statistical Institute, Li Hualei, who's at Stanford, Tiang Xi, who's at the University of Virginia, Anshul Kundaya, he's the only card-carrying biologist in the group, who is at Stanford, and Rachel, as I mentioned, and Yu Ting, who's currently a student. So that's it, and I hope you enjoyed the show.
Days from the University of Connecticut Statistics Department and your host for the remainder of our program. Thank you, Peter. That was a compelling keynote address. We are looking forward to your interview coming up. We have our first poll results. Let's see how many academic descendants you thought Peter has. 5 said under 300, 11 said 300 to 500, 10 people said 500 to 700, and 14 people got the correct results over 700 descendants. So the correct answer was D. Now that we know how many academic descendants Peter has, let's test your knowledge again. The polling window will open up shortly. If you don't see the poll, go to the tab that says Live Polling. And the question is, which of the following awards was given to Peter Bickel? A, COPS President's Award, B, member of the National Academy of Sciences, C, MacArthur Fellows Program, also known as the Genius Grant, or D, all of the above. Go ahead and make your choice. We will share the results of the second poll after our next segment. Next, we have some warm wishes from our generous sponsors. We will be hearing from Demisi Alemeyu from Pfizer and Ronald Wasserstein from the American Statistical Association. Following their messages will be a short break, and then we will resume at 4:05 p.m. Eastern Daylight Time with the interview portion of our program. Let's enjoy our sponsors' videos.
Hello, my name is Demisi Alemeyu. I am head of Statistical Research and Data Science Center at Pfizer. On behalf of Pfizer, it is my great pleasure to congratulate Peter Bickel, my former professor and renowned leader in our field, on his election to be the speaker of the 26th Distinguished Statistician Colloquium, sponsored by my company in partnership with UConn and ASA. Pfizer is exceedingly delighted to be a sponsor of this coveted symposium, which has been graced over the last several decades by some of the greatest names in our profession, among whom were several colleagues and former teachers of Peter from Berkeley, such as Jerzy Neyman, Eric Lehman, and David Brillinger. When the symposium was briefly interrupted a few years ago, there was much concern in the statistics community that would be deprived of the opportunity to honor and recognize our distinguished members such as Peter Bickel. In this respect, we are grateful to Kanan Tarajan, head of Global Biometrics and Data Management at Pfizer, Deepak Day of UConn, and Ron Wasserstein of the ASA, who had the foresight and the vision to reinitiate the series about 3 years ago. At Pfizer, statistics and statisticians play a prominent role, not only in contributing to the company's overarching goal of bringing breakthrough medicines that change patients' lives, but also in influencing the ongoing transformation in our discipline, ranging from the introduction of innovative trial design and analytical strategies to the implementation of state-of-the-science tools in machine learning and artificial intelligence. Pfizer statisticians are especially thrilled to see the recognition of Peter, whose towering contributions have had profound impact on their day-to-day roles of fostering quantitative approaches in decision-making. This, of course, may not come as a surprise to anyone who has followed Peter's research from the early days of his career at Berkeley to the present moment. As a leader of the so-called third-generation statisticians, in the 60s and 70s, Peter ventured in groundbreaking work, including robust statistics, asymptotics, and non-parametric statistics, tools that are routinely used now by pharmaceutical statisticians. Before the bootstrap became fashionable in the 1980s, Peter, in collaboration with the late David Friedman, explored the limits and limitations of the procedure in a seminal paper that inspired generations of statisticians, including myself, both at Berkeley and beyond. As I'm sure we will hear later in this program, his early work was then followed by more profound contributions in fields as diverse as computational efficiency, genomics, weather predictions, and other high-dimensional data analysis problems. Peter is not only a prolific researcher, but also a highly sought-after mentor and advisor. Of course, one doesn't need to be Peter's former student to benefit from his advice and guidance. I remember as a grad student at Berkeley, one sunny afternoon, I saw Peter in the math library at Evans Hall flipping through a book. I casually approached him and rudely interrupted his reading to pick his brain about a question that was on my mind. To my amazement, he dropped the book he was examining and quickly started dissecting the problem. When I went back to my office and shared the experience with my office mate, he reminded me of the well-known fact why every PhD candidate in the department at that time would want to have Peter as their advisor. Indeed, according to the mathematics genealogy project, Peter has over 61 students and at least 700 descendants. Most of his students are now leaders in their own right, working at major universities and other institutions across the globe. In a recent interview with Yakov Ritov, one of his former students, Peter quoted his former advisor, Eric Lehman, who characterized people as either problem solvers or system builders. Peter then went on to say that although he falls somewhere in between, he primarily considers himself a problem solver. That, in essence, is what makes this recognition of Peter even more symbolic and fitting at a time when the world needs problem solvers like Peter, and when more than ever before, the role of statisticians as problem solvers has been fully recognized, especially as we tackle one of the greatest challenges mankind has ever faced, the COVID-19 pandemic. As beneficiaries of the trailblazing work of great pioneers such as Peter, Pfizer statisticians take extreme pride in being a part of the problem-solving team that has brought to mankind the first effective and safe vaccine against the dreaded plague. So congratulations to Peter again on this well-deserved recognition, and thank you for your leadership, mentorship, and impactful research contributions over the years.
Hello, everyone. I'm Ron Wasserstein, Executive Director of the American Statistical Association. I want to welcome you to this wonderful session, this wonderful opportunity. The ASA has been a partner for a very long time in this effort, and we're so delighted to continue to collaborate with UConn and Pfizer, because it's so very important for us to have the opportunity to collect these sessions, these opportunities to talk with, to hear from the great statistical leaders of our time. When I was a young statistician, so long ago that I can barely remember, I do recall how delightful it was to attend a session by one of the senior statisticians, by someone who was well known to me as a grad student. And I remember thinking what a privilege it was to have an opportunity to sit in and hear a presentation by these people. It's so much better to then to not only have the opportunity like we have today to sit in on such a session, but to be able to capture that for future generations so that people can value the work that these people have done and get to know them a little bit as well. That's just so important. And so we're very glad to be a part of that. So thank you for participating. And of course, congratulations to Peter. Look forward to hearing this session. And again, thank you all so